Now first let's start with the innate immunity. So when I talk about innate immunity, let's say you already know that what are innate immunity cells. These are the cells with which we are born. So now point is, how do innate immunity cells identify the other cells? So what are the patterns that they identify? So remember, to understand this, you remember that every pathogen has some molecular patterns on their body, isn't it? So these patterns are called as pathogen associated. What are they called as? Pathogen associated molecular patterns. These are called as pathogen associated molecular patterns. So these are very important patterns which are present on the on a pathogen. So these are called PAMPs. Whereas whenever, okay, a organism or some problem occurs with the cell, okay, so this cell will release certain mediators, like uric acid, okay, or necrosed cells will release certain or some molecules like heat shock proteins will be released. So these molecules will be called as DAMPs, that is danger associated molecular patterns. So it is like as if somebody is trying to catch you, okay, and you will scream. So screaming is a danger associated molecular pattern. So screaming is a danger associated molecular pattern. You understand that? So when you touch somebody and identify that, that is a pathogen associated molecular pattern. When you start screaming, okay, that I am attacked, so that is danger associated molecular pattern. So there are two patterns which could be identified by the innate immunity cells. Either they identify the patterns of the pathogen, that is PAMPs, or they identify, what do they identify? DAMPs, that is danger signal, which are produced by many cells. So these are the two patterns which any cell can identify, PAM and DAMPs. So once we are clear with PAMPs and DAMPs, let us see how do they identify and who identifies that. So every innate immunity cell, so like let's say I am drawing this innate immunity cell. So every innate immunity cell has certain receptors for identification of these PAMPs and DAMPs. So first we are talking about PAMPs, okay. So every cell on its surface has receptors which are called as toll-like receptors. What are they called as? Toll-like receptors. So these are called as toll-like receptors. So these toll-like receptors, they are not only present on the cell surface, but they are also present inside the endosomes. So they are present on the cell surface and on the endosomes. Okay, so toll-like receptors are present on the cell surface and also on the endosomes for catching the pathogen, okay. So now we have approximately 10 toll-like receptor. How many toll-like receptors we have? 10, okay. So total 10, but at least some you should remember, okay, these are mandatory. So for gram-positive bacteria, whenever gram-positive bacteria enters our body, it is identified by toll-like receptor 2. TB bacteria is also identified by toll-like receptor 2. Okay, double-stranded RNA virus. Okay, double-stranded RNA virus is identified by 3. Okay, 4. Gram-negative bacteria. Okay, leptospira, chlamydia. Okay, is identified by 4. Toxoplasma is identified by 5. These are mandatory. If you can remember, 6 is mycoplasma. Okay, 7 is single-stranded RNA viruses. 7 is for single-stranded RNA virus, and 9 is for DNA viruses. So we have 10 type of toll-like receptors, and these are the major ones which you should remember, that what is their function. So these are the major toll-like receptors which identify the PAMPs, pathogen associated molecular pattern. So they identify the TB pattern and say, okay, TB has come, okay, but what will they do after identifying? Okay, so chlamydia pattern they can identify, toxoplasma pattern they can identify. What will they do? Once they identify a pattern, okay, once they catch a pattern, they will do two things. First, they will activate NF-kappa B. This NF-kappa B will go and activate the other cells. This will go and activate other cells, okay, other immune cells. They will activate the other immune cells. And second, what will it do is that it will release interferon 1. So either it will release interferon 1 or it will activate the other cells. So major function of these cells is to just signal that somebody has come, please get alerted, and it will try to stop it in the meanwhile by releasing antiviral interferons like interferon 1s. So this is how a toll-like receptor acts. Now let us see what are the other receptors which we have, okay, apart from this. So this is the first receptor to identify PAMPs. Now let's look at the other receptors, okay. Similarly, after toll-like receptor, in the cytoplasm of the cell, we have node-like receptors. In the cytoplasm of cells, we have node-like receptors. Now, these not only can identify PAMPs, so any organism entering into the cytoplasm of the cell could be identified by node-like receptors, or second, any DAMPs, any danger associated molecular pattern can also be identified by node-like receptors. So node-like receptors can identify both PAMPs and DAMPs, okay. So they can identify some pathogen patterns also and danger patterns also, okay. Your body is getting infected by something and body starts releasing uric acid. This uric acid is identified by node, okay. So danger signals are identified by node apart from PAMPs. That's the uniqueness of node, and this node-like receptors are present in the cytoplasm of the cell. Now let's see what happens when they are activated. Once these node-like receptors are activated, I hope you remember we have studied about them already in cell injury. Once these node-like receptors are activated, they will activate inflammasome. Okay, this inflammasome will go and activate interleukin 1, that will cause fever, or they will activate caspase 1 and 11, and that will cause apoptosis. So this together, do you remember the phenomena that we have studied? I hope you are remembering this. This is called as pyroptosis. What is this called? This is called pyroptosis. Pyrexia associated with apoptosis. Okay, this is called pyroptosis. Is everybody clear? So pyroptosis, when I talk, or apoptosis, is done by node-like receptors and they kill, okay, the pathogen. So I told you, any innate immunity receptor, either it will go and activate the other immune cells or it will cause apoptosis. So can you see here the cell undergoes apoptosis. Here the cell undergoes apoptosis with that pathogen, okay. Now once we are clear with this, now here I want to tell you a disease, okay. So this, node receptors are very, very important. Now they can cause fever. So now I will be teaching you diseases side by side so that you can remember them side by side, okay. So gain of function, mutation of node, okay, node receptors. If there is a gain of function of node, okay, especially node 2, gain of function of node receptors, if there is a gain of function, they become activated even without any injurious foci, node is automatically activated, then what is going to happen? There is going to be lot of pyroptosis. So this causes what is called as lot of familial fever syndromes or periodic fever syndromes. So this causes lot of periodic fever syndromes. Okay, so periodic fever syndrome. So patient is going to get lot of fever syndromes. So gain of function of node 2 mutations typically causes periodic fever syndromes. So they are periodic fever syndromes. Not only that, remember in inflammatory bowel disease, node 2 mutations are very common, especially in Crohn's. Okay, in Crohn's disease, node 2 mutations are very, very common. I'll be posting that in GIT also here. Also, you can see my YouTube video on inflammatory bowel disease where I have already told you that node 2 mutations are very common in Crohn's disease. I hope you understand, because they start responding to innocuous antigens. Even without antigen, they start reacting, and this causes autoimmune diseases. Okay, so Crohn's and periodic fever syndromes are mutations of node-like receptor. Okay, now once we are clear with this, now let's see what are the other innate immunity receptor. Apart from these, 2 more receptors have been recently identified. Okay, 2 more receptors have been recently identified. In the cell membrane, they identify C-type lectin receptors. C-type lectin receptors. So C-type lectin receptors are present in the membrane, and Rig-like receptors. Rig-like receptors are present in the cell cytoplasm. So we have 2 new receptors, C-type lectin receptors and Rig-like receptor. Okay. Now, C-type lectin receptors are basically meant to identify fungi. Okay, so whenever fungi enters into the cell, that is identified by C-type lectin receptors, whereas Rig-like receptors are used for identifying viruses. So now here is the catch. Whenever the viruses are present in the cell surface or endosomes, then they are identified by toll-like receptors. So we have various toll-like receptors for viruses, like toll-like receptor 3, toll-like receptor 7, toll-like receptor 9. They are for viruses. But then virus should be identified at the cell surface or endosomes. If the virus has come in the cytoplasm, okay, this is usually identified by Rig-like receptors. So Rig-like receptors are usually meant to identify viruses which have entered into the cytoplasm, whereas surface or endosome is by toll-like receptor. Fungi is by lectin receptors, okay, and nod-like receptors, DAMPs, especially for DAMPs. Is everybody clear? So these are your innate immunity receptors and how innate immunity works. Now once we are clear with innate immunity, now let's come to the adaptive